Hello everyone, my name is Naorem Jemes Meitei, and currently I'm working as a PhD student at the Kaohsiung Medical University, Taiwan, in the Department of Medicinal and Applied Chemistry. And today I'm going to share you some of the work which I'm working on now, how the nitrite get reduced to release NO, and what is the mechanisms lying on it. So before I start sharing my work, let me introduce how the people get attracted to such kind of works in these nitrification steps. There are a lot of steps like this, nitrite oxidation, dissimilation, denitrifications. So in this, we focus in our lab that we focus on these particular steps where the nitrite get reduced to release NO, which is one of the most important steps in these nitrifications. So as per reported by the J.W. Golden in the 1991, they report the crystal structure of this type 1 copper center and the type 2 copper centers, which they found this from the ectodermal vector and cyclotest text. In this crystal structure, they found that the type 1 copper center act as an electron donor site and the type 2 copper center act as the nitrite reductase enzyme site. So basically, if we need to understand how the nitrite get reduced to release NO, we need one electron and two proton source. So produced NO have a lot of applications like apoptosis, vasodilatations, and neurotransmissions, etc. And as we need to understand what are the applications of it, and if there is a lack of production of this nitrous oxide in our human bodies, what are the consequences? So as per reported by the WHO in 2021 and 2023 data, almost 7.25 million and 10.85 million and 19.41 million were passed away due to the total stroke, hypertension, or the cardiovascular disease. So in order to understand and in order to find out a drug to save millions of people, we synthesize our beta-diketonated copper to nitrite complex as a NO source, and we try to mimic these beta-diketonated systems through this biological system, which the 3 histidine binds in this type 2 copper center, and in our system also there is 3 nitrogen bind in our copper 2 systems. So after synthesizing this copper 2 system, we study by using these different kinds of thiols, and we also use these biologically active thiols like L-cysteines as an electron and proton source. And after reacting with these different kinds of thiols, we found that the NO is released from this beta-diketonated systems as 100%. So after finding that the nitrite get reductions, and we understand that when we investigate the mechanisms lying on it. So in order to study these mechanisms, we propose the 3 mechanism pathway. The first one is oxygen atom transfer, the second one is nucleophilic attack, and the third one is the acid-base actions. So after studying the investigations, the careful investigations, and through different spectroscopic technique, and we propose that it follows through this acid-base actions pathway, which no one ever reported before. And we also rule out the oxygen atom transfer and the nucleophilic attack. So for the further details for these mechanisms lying on it, see you at the May 16 and the NTU at the Merck Young Scientist Award 2026.